Hi everyone, so we're looking at D.5, and in this topic we'll be looking at bacterial and viral infections and the difference between bacteria and viruses. So this is what a bacteria generally looks like, and bacteria are single-cell organisms. Bacteria are self-reproducing units. Here we use the term reproducing, and bacteria, they do not need a host cell to reproduce. They will reproduce by themselves, and inside the bacteria cell you have organelles or subunits. So you have a cell wall, you have DNA, you have a cell membrane, plasma membrane is the cell membrane, you have a cytoplasm, and inside you have organelles, and they will carry out metabolic processes. Metabolic processes include respiration, reproduction, growth, etc. So these metabolic processes means that bacteria behave as living organisms. Now, viruses, on the other hand, they are smaller than bacteria in size, and they need the cell of another organism in order to replicate. Here, I'm not using the term reproduce, it's replicate, because viruses will make copies of themselves, and they will use the living cell of another organism. So you can see here, this is how viruses will replicate themselves. They will inject their viral DNA or RNA inside another cell and will cause the cell, as you can see here, to make copies of itself, and then it ruptures the cell, and then the viruses will go out. So this is called replication, and it needs a host cell to do it. Viruses do not carry out metabolic processes. They do not feed, they do not grow, they do not undergo excretion or respiration. So these are metabolic processes. These are processes that allow us to classify as something as being a living thing. So we consider viruses to be non-living thing because they will not carry out these metabolic processes. Their aim is only to replicate themselves. So they are complex chemical compounds, and we can synthesize viruses in laboratories. We can also isolate viruses in crystalline forms, and viruses will derive any energy that they need from the host cell. So when they are outside living organisms, they do not perform any biological process. It's only when they are inside host cells that they can do that. Now, viruses have two main components. One is a coat, which is called capsid, as you can see here. So you have a protein coat, and then you have DNA or RNA. DNA is deoxyribonucleic acid, and RNA is ribonucleic acid. And both of these are nucleic acids. And these nucleic acids, these are genetic material. And viruses contain a simple version of genetic material, and this is what is going to be used by them to make copies of themselves, right? So, as I said, viruses do not exist outside living organisms. Outside living organisms, they will not carry out any biological function, and when they hijack the metabolism of a host cell, then they can make multiple copies of themselves. So how do they make copies of themselves? The process is described here. So let's go through this. So as you can see here, so the first step is the virus will attach to the surface of the cell, of the host cell. So it will attach to the cell surface. So what happens is that the capsid protein is going to bind on receptors found on host cell surface. So if we have to explain what are receptors, so here we have cell membrane as number 2 here. And inside the cell membrane, you have, these are receptors. And receptors, they have specific sites where they can accommodate specific molecules. So you can see this one has a triangle shape and this one has a circular shape. So specific molecules can fit in specific receptors, and so the virus will bind to one of these receptors and is going to send in the nucleic acid, RNA or DNA, inside the cell. So virus will stay on the membrane and is going to inject the viral DNA or RNA into the cell. The coat of the virus will remain outside. Now, the material that goes in, the DNA or the RNA, is going to be interpreted by the host cell as an instruction to make copies of the nucleic acid. You can also see here you have new protein coats which are synthesized inside the host cell. And we have the protein coats and the nucleic acids are then assembled in step E here, and you have mature viruses which are assembled inside the cell. And once the viruses are assembled, you have the rupture of the cell membrane of the host cell, and then you have the viruses that is going to be released. So you have examples of viral infection: smallpox, chickenpox, cold sores, AIDS, influenza, the common flu, polio, measles, rabies, and also COVID-19. Now, here you have a table that shows the differences between bacteria and viruses. Bacteria are self-reproducing, viruses need a host cell. Bacteria are able to carry out metabolic processes, viruses do not. Bacteria have organelles. Viruses only have a protective coating and genetic material. This is the DNA or the RNA. Bacteria are larger in size, viruses are smaller than bacteria. Bacteria have more complex DNA. Viruses will have simple DNA, or they will have either RNA or simple DNA. And bacteria, they multiply much more slowly than viruses, while viruses will multiply really quickly using the host cell. You need to know these differences.